Thank you. So the Cape Fear River Basin is home to 5 native bullhead species, including white catfish, and 3 native mad toms. Channel catfish, blue catfish, and flathead catfish were stocked throughout the 20th century by various management agencies. Most of the catfish research in North Carolina and in the Cape Fear River Basin is focused on flathead catfish. Diet studies and trophic dynamics like David just presented, also population dynamics and telemetry. Very little published research has actually looked at the Ictalurid community as a whole. One notable exception is the Moser and Roberts contribution to the Catfish 2000 proceedings, who determined that native Ictalurids were pretty much extirpated from the Cape Fear River Basin main stem, main stem Cape Fear, Northeast Cape Fear, and Black Rivers. But their study was kind of limited just to those main stem areas, and their surveys were just conducted in the 1990s, although they did look at some kind of fragmented surveys going back to the 50s. The Wildlife Resources Commission has a pretty, very long history of sampling fish going back to the 1960s, and my goal was to merge all of our legacy data with contemporary surveys to really explore catfish trends through time, and not just in the main stem larger tributaries, but in all the tributaries in the basin. And I failed to mention, I'll pretty much be focusing on the Cape Fear River Basin downstream of Buckhorn Dam, delineated just in the dark areas of the basin. Upstream in the Piedmont sections of North Carolina is highly fragmented. There are a lot of municipal reservoirs and a large Corps of Engineers flood control reservoir. So all the fish sample in that area is pretty much in reservoir fish communities. Most of what I'm presenting is all riverine. We have a considerable amount of electronic data available to us. Our agency conducted a baseline rote known survey in 1962 that had a lot of sampling events really throughout the basin, a lot of both main stem and in smaller tributaries. And that data set was digitized several years ago. We also have a sport fish database that went online in 2010 and contains some legacy data that is uploaded as staff schedules allow. Our Aquatic Wildlife Diversity Program has a database that was initiated in the 1990s, and that program focuses on species of greatest conservation need. Primarily, that's conducted in wadable streams, but not exclusively. We also have special projects like the hand crank electrofishing project that Michael Fisk presented before lunch. But 98.6% of the data that we have available electronically was either those surveys conducted in 1962 or since 1992. So there's a really glaring 30-year gap in our available data set. But we had 2 filing cabinets chock full of paper data sheets that contained a lot of rote known and electrofishing surveys throughout that time. And a goal of mine was to get that and to be able to merge that into our electronic record. This is an example of a typical paper data sheet in our archives. This would not have been the actual sheet that was recorded in the field. You can imagine field surveys are getting pretty grimy and dirty, but this would be a summary data sheet that was compiled afterwards, and it contains all the necessary information to be able to figure out how many fish were caught where and when and by what gear. And we're also fortunate to be able to talk with the agency staff, and in many situations be able to talk with agency staff who actually did conduct those surveys 30 and 40 years ago. Overall, after getting all those surveys merged into the electronic record, we had over 928 sampling events conducted from 1962 till 2019. There were actually over close to 2,000 sampling events, but the difference there is all targeted surveys were removed, so surveys that were just for largemouth bass and sunfish, or we have a lot of anadromous surveys just catching shad and striped bass. So all those targeted surveys were removed, and those were identified based on either the catch or also confirmed with conversations with the biologists who conducted those sampling events. Those 928 sampling events caught a little over 16,000 catfish. Merging the paper archives, the paper archives contributed 20% of the total number of sampling events, and 36% of the actual catfish that were caught, and that filled in that 30-year time gap that we had missing. All of the sampling event data I imported into QGIS and merged those sampling event locations with the NHD Plus data set. And if you haven't used that before, that's a really awesome data set of stream data. And I merged several environmental factors in that data set, including Strehler order at each sampling location, mean annual discharge, mean catchment temperature at a sampling location, mean water velocity, and so on. I also conducted a stream network analysis, and I was interested in the distance of each sampling location with each of 3 known flathead catfish stocking sites in the 1960s. And I'll expound upon that in just a minute. After this, I used the vegan package in R to conduct non-metric multidimensional scaling to just help visualize environmental gradients and community patterns. This is the mean catch per sampling event through time, and the black indicates native catfish species in aggregate, and the hashed is non-native catfish species. And it's apparent that from the 1960s through the mid-1990s, the catfish community was dominated by native species. Since the late 1990s, really we've just seen it flip, and really non-native catfish species, blues, flatheads, and channels are dominating the catch. There is a pretty clear abundance shift. The 1960s through the 1980s, we conducted a lot of rote node sampling. We began electrofishing in the mid-1970s, and really that's been the predominant sampling gear since the early 1990s. This contains a lot of different sampling gears, so really the mean catch per sampling event's a little fluid, but the next several maps are all going to be pretty similar, and I'll just explain this in a little bit. But the dark blue indicates that the percentage of the total catfish community within a 10-digit USGS hydrologic unit was 75% or greater were native catfish. Going through lighter shades of blue, declining contribution of native catfish to the total catfish catch until you get to white. Within the basin, white denotes that fewer than 25% catfish were native species. The 3 point markers are the known flathead catfish stocking locations that were stocked by our agency in the 1960s. The one that is most known was in 1966. This is the stocking of 11 flathead catfish adults at Tar Heel, North Carolina on the main stem Cape Fear. But there were 2 stocking events the previous year of juvenile flathead catfish. One in Lake Rim, which does have an eventual surface water connection to the Cape Fear River, and about 100 juveniles were stocked in the northeast Cape Fear River that same year, 1965. So in the 1970s, the one additional color, gray, gray just denotes that there were no sampling events within a 10-digit HUC in that decade. But we do see, compared to the 1960s, we are starting to see fewer native catfish in some of these watersheds. 1980s, a large spatial component that was not sampled, but there was intensive catfish sampling in the upper watersheds of the Cape Fear, and also in the lower Cape Fear and the northeast Cape Fear drainages. 1990s, 2000s, and the most recent decade, and there's in no watershed have we seen the overwhelming majority of the catfish, actually be native species anymore. This map is a little bit different than the previous ones. This denotes how long it's been since we've seen a native catfish, any of the native catfish species within a 10-digit watershed. And the dark blue means we've seen them within the last 5 years. This light blue color, it's been 5 to 15 years since a native catfish has been observed. White means it's been greater than 15 years since a native catfish has been observed. And there's been intensive sampling in a lot of these wide areas. The point markers denote the actual sampling event and when it was conducted. White is in these white circles are in the last 5 years, but it's kind of difficult to see here. They're pretty, our sampling's pretty well distributed. Here are the native catfish species in the basin, along with their last observed location and year. Several of these species we have seen fairly recently. Tadpole mad tom and brown bullhead. It's getting close to 10 years since they were last seen in the Black River watersheds. But white catfish and broadtail mad tom were getting past 10 years, 20 years for broadtail mad tom, which is actually an undescribed species that appears to be genetically unique from other populations of broadtail mad tom. It might be a distinct species unto itself here. I have an asterisk by white catfish because David Belkowski actually saw a white catfish in the lower Cape Fear River last year. But this occurred after about 6 months of extremely high flows after Hurricane Florence. And in the upper Cape Fear River Basin and Jordan Reservoir, there is a viable white catfish population up there. And we know that we get a lot of tagged striped bass that were stocked in that river. We see those downstream in that same area. I have very little doubt in my mind that that is where that white catfish came from, that there's not a functional population in that area anymore. This is the ordination from the non-metric multidimensional scaling. And we see the point markers, those are each individual sampling event. The dark point markers are sampling events from the last 30 years. The white are from the earliest sampling events, the 60s through the 1980s. And we see that the non-native catfish species, channel cats, blue catfish, and flathead catfish were actually came out right on top of each other. But white catfish, snail bullhead, flat bullhead, brown bullhead, yellow bullhead, tadpole mad tom, and margin mad tom. I had to remove broadtail mad tom from this analysis because there were so few that had been caught that it just kind of blew everything up. But we do see some structuring in the ordination. In vegan, you can fit you can fit vectors and factors into your ordination. These are the 95% confidence ellipses for the decades. We see the 1960s are pretty much grouped up right with those native species. As you move into the 70s, 80s, and 90s, you're moving closer to those non-native catfish. The 2010s were actually overlapping, doesn't seem to be all that much different from each other, so perhaps by the 2000s we've reached this new non-native catfish equilibrium in the basin. Here's the other environmental vectors from the QGIS and the stream network analysis. And as stream order increases, we see more channel catfish, blue catfish, and flathead catfish, more of those non-native species. And of course, that arrow extends the same direction in distance opposites. As stream order decreases, you get these smaller streams, you start to see perhaps more non-native species. Temperature was kind of the least influential, although as stream temperature decreases, mean annual stream temperature decreases, you're more likely to see those native species. The stream network analysis, so the distance from the flathead catfish stocking locations. Tar Heel, this is the adult flathead catfish stocking location. As you get further and further away from that initial adult stocking location, you're more likely to see the non-native or the native species. It's interesting, the minimum stocking distance. So the closer you get to any of the stocking sites, was associated with white catfish and snail bullhead. And I think what happened was that in the northeast Cape Fear River in the 1980s, adjacent to one of those flathead catfish juvenile stocking sites, a lot of white catfish were caught at that location. Perhaps that signifies that juvenile stocking event in the northeast Cape Fear River really didn't take hold. It was the stocking of adults in Tar Heel that drove the flathead catfish population. So just to kind of wrap up, very few native species have been seen in any kind of recent memory in any of our surveys. And we might be looking at extirpation of broadtail mad tom and maybe even white catfish, which were, historically, that's the large native catfish in the basin. Luckily, they are holding on in our Piedmont right now, where the flathead catfish have been more recently starting to move up into, so they're kind of coexisting at the moment, but we have other river basins like the Yadkin chain of reservoirs where we don't see as many white catfish either. So it remains to be seen kind of how the upstream parts of the Cape Fear River Basin, how those white catfish populations hold out. Broadtail mad tom, things aren't looking very good. They're difficult to catch, but still it's been 20 years since we've seen them, and there have been targeted surveys for those very recently, and they haven't turned up anything. Dave kind of mentioned it, and Corey gave a talk earlier, a couple rooms down, on the North Carolina Catfish Management Plan that we just completed last year. The Cape Fear River Basin is in the what we're calling our invasive catfish harvest unit, and this signifies that basically non-native catfish, we're not going to have any restrictions on harvest throughout the basin. And we're going to, our native catfish species, we are proposing to make inland game fish that actually prohibits the sale of these bullheads and puts a creel limit into place for them. But it might be a little bit late for that because it's hard to see some of those species anymore. If we think about the change that we've seen just over the last 60 years in our survey, it's hard to imagine, but it's kind of easy to imagine looking at the data what kind of change we're going to see in the next 60 years. How easy it is to move fish around, but also in the news we're seeing a lot of the effects of climate change nowadays. Now is the time to really start long-term surveys using a standardized sampling plan, so that you don't have to resort to this kind of backwards look at a lot of disparate data and try and pull something together. But we do have legacy data, and no, it's not just our agency, but there are a lot of agencies out there, a lot of university programs that have paper data sheets rotting in file cabinets somewhere, and now's the time to get those updated into a format that's available to use, because there is useful information in that. I have a lot of current and former Wildlife Resource Commission staff to thank. Even more than this, that actually conducted the surveys through time. But with that, I'll take any questions.
We do have time for questions.
Yes, sir.
With the invasive catfish on the Atlantic slope, especially like in North Carolina, and this may be a question Tom or somebody else can answer, has there been any thought to look at the cascading effects it could have when they start to extirpate native catfish or maybe other native taxa, the endemic species, what it might have like on the freshwater mussel pond? Has that ever been addressed? I'm a mussel biologist.
Yeah, that's a great question. It's something that we did identify in our catfish management plan, was the potential for a couple of different things, not just direct predation, but even our native catfish species potentially being hosts for some of these mussels. We don't have great data on some of those, but that is something that we are considering. I don't think we have looked at a lot of that research, but there's certainly a lot of mussels are more imperiled than our fish species, unfortunately. So, yeah. Great point. Yes, sir.
Just quickly, so you mentioned that the catch period effort changed across time. I was wondering, when you were doing your analyses, how did you account for that so that didn't drive all your relationships?
I didn't. But yes, no, that's a good point. Definitely we caught a lot more species, or not just species, but the abundance was a lot higher whenever we were conducting our rope nose surveys compared to electrofishing. But I didn't partition out anything as far as, through time trying to constrain the different surveys. I know in using CCA you can do that, but I wasn't aware of a way to do that within NDS. But I'll talk to you afterwards if you know the way to do that.
All right, thanks, Kyle.
All right, thank you.